I will tell you a little bit more about it as we go along. So the things to remember here is that this is a biotechnology company, and I've been working on the technology, which is called DARK, for the last 10 years. We've already been able to go as far as phase 2. We finished phase 2 clinical trials recently. And this was because we are funded through the Wellcome Trust, and investors are over £4 million. And so far, we have done 132 patients. So the key thing to DARK technologies consists of two things: a patented biomarker and artificial intelligence. So at the moment, we're a very new company. We've had some seed funding, £500,000 earlier this year. We're at the stage now where we are looking for Series A, £5 million, to allow us to go out and start the business, which will be really almost as an AI CRO, which is we'll be going to our pharmaceutical company partners, trying to test their drugs and get revenue in that way. So the DARK technology, and I'll show you a picture about that shortly, has already been recognized by regulatory authorities, that is the MHRA and the FDA, as an exploratory endpoint. And this is really important because in the diseases we're talking about, and these are chronic neurodegenerative conditions, there is very little out there that can give you a quick and effective idea of whether your drug is going to work. So if you can de-risk that, and we believe our technology can help doing that, then that drastically reduces not only the cost, but the duration of your clinical trial. So initially, we will be focusing on glaucoma and AMD, and that's not just because I'm an ophthalmologist, but it's because we have patients that have already had the intervention. But we will be expanding to Alzheimer's disease and Parkinson's disease and multiple sclerosis. And we have very good preclinical evidence of that. It's all been published, where we have shown that the technology is very effective, both looking at the disease activity, but also as a response to treatment. So to date, we've had around 12 pharmaceutical companies who've come to us asking us to see if we can test their drugs. And as I said, this is primarily in glaucoma and AMD, but these other areas are actually looked at. So over here on the right, that is the retina, the back of your eye. This is when you go to see opticians or your ophthalmologists, they look into the back of the eye, and this is what they're looking for. But what you see here that you don't normally see are the white spots. Can you see the white spots in that greyish background? Now, those white spots are individual nerve cells that have been labelled by our biomarker. So the biomarker works by picking up cells that are either stressed or in the stages of apoptosis. Apoptosis is a form of cell suicide, and it's a very good marker of disease activity. We know that in glaucoma and in AMD, you have apoptosis in the key cells, in the retinal ganglion cells in glaucoma, which you've already heard a lot about from Max, but in the photoreceptor cells in age-related macular degeneration. Now, where you may not have been aware that the retina is basically a window onto the brain. And the same cells that are in the brain are also present in the retina. And it's become increasingly recognized that if you have an abnormality in the brain, you will probably pick up something in the retina. So for example, in Parkinson's disease, optical coherence tomography, which is the thickness of the retina, is already being used by some people as a biomarker to test the efficacy of new anti-Parkinson's drugs. We believe that the DARK technology will not only help in a clinical setting for testing a new drug, but we've already shown over the last 10 years that it is very effective at looking to see if your candidate drug is effective in a preclinical model. And moving forward, the most important thing and the reason why we're saying this company initially is starting off as a biomarker is because it can predict those patients at higher rates of progression. So this is just a little cartoon showing you the mechanism of action. A lot of you may be aware that when a cell is under stress, and particularly when it goes into apoptosis, you have a translocation of a molecule called phosphatidylserine from the inner cell membrane to the outer cell membrane. And the marker that we're using, which is annexin, just has a high affinity for sticking onto this phosphatidylserine. Now, taking advantage of the fact that the eye is transparent, we fluorescently label the annexin, and so you can pick up those little white spots that I showed you in the previous picture. So the actual process of binding of the annexin not only shows you if the cell is in apoptosis, but also if it is stressed. Now, what we've been doing with our artificial intelligence is that we've been looking not only for an objective measure of the number of cells and the types of cells, but the pattern. And the pattern is really what we believe pulls apart the different diseases. So I mean by that in glaucoma, when an ophthalmologist looks at the back of your eye, they look for a pathognomonic change in the nerve. It's called an optic disc cupping, something that you see when an ophthalmoscope is used to identify whether your patient has glaucoma or not. With age-related macular degeneration at the macula, you have all your pathology. So the spatial distribution of these white spots will give us a very good indication of whether it fits into those two diseases. And we're hoping as we get more and more patient data coming through, we'll be able to look at the AI elements for the other diseases as well. So what I mentioned that we can predict those who actually progress rapidly. It's very important in our first use of this. So with drug companies, one of the big things is patient stratification. So the first thing we are suggesting is that we can enrich the patient cohort by using DARK first as a triage way of going for those patients who have the highest rate of risk of progression. So once you have those patients, you can treat them with either placebo or your drug, and then be quite happy that that will give you the greatest chance of showing whether your drug is effective or not, because you're going for the high-risk patients. Therefore, if your drug works, it's going to work in that group. The importance also is we've already shown that the dot count predicts changes in retinal nerve fiber thinning and visual field by 18 months. So it's 18 months ahead of what we see in the gold standards we are picking up in the retina at the time. And that means that when you're talking about visual fields, which normally take 18 months to 2 years before they're showing a change, we're talking about weeks. And that is why we're saying that this reduces the risk and the cost and the duration of your proof of concept trial for looking at a new drug. So here are some figures here showing you what we are predicting. This is just the biomarker in macular degeneration and glaucoma. So we know that there are 83 trials planned currently in those two diseases. Roughly, that's 29,000 patients. If we are charging 16,000, that's with obviously our profit margin in there, that is 500 million pounds. So that's our initial market that we're going out for. But we're not just a one-horse race. We have other assets. These are some drugs that my team have developed. This is all through UCL. So the first one we've already shown to be neuroprotective in glaucoma and Alzheimer models preclinically. We have a similar drug, which is also going through the same process. And we've also got a third diabetic drug, which also has a neuroprotective effect. And not on here is an option on one of my other technologies, which is an eye drop treatment for macular degeneration, eye drop of anti-VEGF. So I hope I've summarized some of the things that we will be covering with the company. I think one way of regarding our platform initially is that we're talking about it being an AI CRO, and we're obviously going to be almost acting like a service company, because we will be allowing drug companies to use our biomarker to test their drugs. Initially, we're going out for glaucoma and AMD. And as I said, we've already had approval by the FDA and the MHRA for this to be used as an exploratory endpoint. Key is our IP. We have patents. In fact, we have 4 patents now, 2 on AI, 1 on the actual annexin protein, and another on another version, which I have forgot to mention about. So the phase 1 and phase 2 trial involved an intravenous administration of this biomarker. We've actually developed a nasal inhalation version, which will make this a non-invasive test completely and allow it to be accessible to the high street optician and technician. So we think we are a disruptive technology because we're one of the few technologies that you can see single cell disease. And we believe that this has widespread applications. We're talking about Alzheimer's, multiple sclerosis, and Parkinson's. We are in the midst, in fact, we've just submitted our first paper from the phase 2 trial, and the results will be very shortly. Of course, under a CDA, we're very happy to share that with you. But for the moment, what we're doing is we're looking for Series A £5 million funding, and really, I'm very happy to answer any questions if you have any. Thanks.